I'm going to start by explaining why we need databases, why we need to integrate data. And I'm going to move on and talk about how we express that data. And then finally, I'm going to give you some examples about how you can find useful information, which I guess is what we are going to be most interested in. But I think it's important to understand the basics as well, some of the basics at least. And then I am going to do a little bit of systems biology at the end, just to please Glauco, but just a little bit.
Very end.
Yeah, just a tiny bit. So in many ways, the web is like a library. So, you know, we all use the internet now like we used to use libraries in universities. And there are features of libraries which we should recognize. So the books are structured, the way that the books are organized are structured. They're searchable after a sense, so you can look at them, you can find things in indexes. They should be up to date, although of course books are not as up to date as the internet. And there are references between them, so you can find in one book where that information might have come from. And a lot of that applies to data too in the modern world, but there are other features as well. So partly because of the automatic nature of much of computation, we can have summaries, we can have all sorts of different kinds of data, so we can have numerical data, we can have images, all of which are live objects in a sense. And we can have, you know, quite specialized data types like MRI images, fax scans, that kind of thing. And the other thing that we can have on the internet that you can't have in books are tools to allow you to analyze that data. And that's something that you would never had, you know, just by going to the library. So the world of data is much expanded now that we have the internet. And so that clearly it's a useful way of accessing information, but there are other reasons as well why we need to develop repositories on the internet to make data more accessible. And one of them is the fact that there's a lot more data now than there was 10 years ago or 20 years ago. So, for example, the European Bioinformatics Group Institute, which is at Hingston near Cambridge, is a large centre, obviously, for biological data. And it holds 6 of its own core databases, which are seeing exponential data growth. And I'll show you a bit about that in a minute. A lot of collaborative projects are based there. In fact, very often nowadays, large projects in Europe almost have to have the EBI as a collaborator as part of a project, if they're doing informatics, and nearly all projects need informatics. So their website has over 3 million hits per day. They have a million unique users a year, so there are obviously a lot of scientists out there. And, you know, there are various other databases also held at the EBI. And so these are some images of data growth in different types of data. So that's the Int'l sequence bank. You can see measured in megabases the rapid expansion of data. And virtually all data types show the same kind of expansion. So there's massive expansion of data. And the more data there is, the more difficult it is, obviously, for anybody to individually deal with this. So when I started doing computing in the mid-80s, you couldn't exactly print out all of the Int'l database, but you could have an idea what was in the Int'l database. Now it's massive, and you can't do that. You need more tools and more sophisticated ways of dealing with the data. And as part of that, of course, is the mouse, and the mouse has been expanding. And this is quite a nice image, in fact, of how important the mouse is becoming. So this is just based on PubMed. And it's the number of publications referencing different model organisms in recent years. This is actually a little bit out of date now, but it shows the trends. So the blue line is mouse, the red line is rat. So the rat is traditionally a physiological model, of course. And then these are the other model organisms, so yeast, C. elegans, zebrafish, and Drosophila. And you can see that the mouse is the big model organism now. And so we need to think carefully about how we reference that data, particularly because, of course, we want to be able to reference the data between mouse and human. And here are some examples, some more specific examples of the same kind of analysis. So if we want to look at cancer, or we want, you can see the mouse as the leading model. It hasn't been quite so true in some other general areas. So, for example, in diabetes, rat has been the major model, but mouse is catching up fast. Roger is pushing that as hard as he can. Yeah, it's all his fault. And heart disease, the gap is closed. So the mouse is getting bigger and bigger as an area of research. And in specific areas, specific disease areas, is quite clear. So this is muscular dystrophy. The mouse models are obviously the most used in Alzheimer's and in neural tube defects. So these are just some examples of disease-related areas. And you can see that the mouse is a big organism. So my own perspective over the last few years has been very much working on the storage and integration of mouse data. So I'm going to work towards telling you about that. And I should have said at the beginning that a lot of these slides are inherited from my colleague Paul Schofield. So if I pause briefly at some point, it means I'm trying to work out what Paul wanted me to say at this point. But I think I've got the general drift of it. So the point about databases is so you can share your data in different ways. So you can have data on your website. You can do an experiment. You can put it into a paper. You can put that paper up as a PDF. You can put data up as a table on your website. But that's only a very limited way of sharing data. It relies on people being able to make use of what you've given them and understanding what you've given them. And really, what databases are about is making that as transparent as possible and making the data as easy to access and understand as possible. So this is particularly important now because a lot of public money is going into research, and the funding bodies are realizing that they really need to. They're not just going to give scientists money to do research anymore. They want something out of it at the end, and they want that data to be out in the public domain so that all the scientists can make use of it. This is very much like what Wolfgang was talking about earlier. This is pre-competitive in the sense of it's much more useful for the scientific enterprise if the data is out there freely available and usable than if you just publish it and nobody sees it again. Once everybody has access to all the data, they can do a lot more with that data, and science progresses faster, at least that's the theory. So the aim of putting data into databases is to maximize public benefit of research and to drive this cycle of discovery. And that means that the data is somewhere long-term. So it's not just published and then disappears. It's somewhere where you can access it for a long time. You can look at different data sets which relate to each other and try and compare that data, which again would be difficult to do. And it applies not only to data, but also applies to objects. So again, Wolfgang's description earlier was of databases which give you access to ES cells or to mice, and we'll come back to that later. So again, you need to have places to get access to that material, and you need to avoid as much as possible duplication. So you want to know, is that already available? Should I make that again, or is there a need to make that again? So all of that is important, and it's both good for science and good for the funding bodies. And in particular, of course, very large-scale data-generating projects. So for the obvious example being the Genome Project, need to have somewhere to disseminate their data, because you can't, download a genome sequence and make sense of it yourself. You need somebody else to do that for you, and that's part of the need for all of this. There are other areas, there are other sort of related issues. So one of them is accountability. So this is a bit of it's a last year slide, but it's still quite relevant. So this is the famous case of the climate change data, which that was being accumulated over a long period of time. And what had happened was that the data, the raw data, was no longer available. So it was published, summary data was published, but the raw data was no longer available. And this meant that people couldn't replicate the analysis. And this is particularly in something as sensitive as climate change, very bad news. Data has to be available because reinterpretation is always possible. And so there's a
problem is to decide what you want to keep.
Yeah, that's true. So there's a pressure to keep all the data, but there's also pressure not to fill up every hard disk on the planet. So there's this so famous 280 exabytes of data or something that are now currently available, stored on the planet, and that is an issue. And I probably don't deal with that, so I can talk about that now. So, for example, with genome sequencing, so now we have a 1,000 genomes project in sequencing 1,000 human genomes, which will be more than 1,000 genomes. Do you store all of that? Well, do you store all of that data? Do you store the raw data, all the sequence runs? Do you store the entire genome sequence, or do you just store where that genome sequence is different from a reference genome, which is actually the rational way to do it? So thinking about what you store and how you store the data is also important. And then there are other issues which we're faced with. So we're quite used to dealing with sequence data. We sort of know what that looks like, and we know a bit about what we can do with it. But there are constantly new kinds of data coming out in large quantities. And sometimes the bioinformatics is behind the wave of these new data types. So there's a constant need for new databases dealing with specific kinds of data. And there's a constant need to bring those new types of data into the larger international fold of databases. So you want to go to a genome browser and find as much as you can about a particular gene. So you want to have access to pointers to all the different kinds of data that might be out there. So there are lots of things that need to be dealt with, if you like. So I'm going to start at the bottom of that pile, start with data sharing. So this is something that came very much out of genome projects. So when you may remember, some of you may remember the controversies at the early stages of the human genome project, where there were competing groups of people. There was a public consortium and there was a private consortium. The private consortium was using public data to compile their own version of the human genome, and this was behind a license wall, and you couldn't get the data unless you paid to get the data. And this gave rise to a significant backlash in the scientific community. Why should we pay to access something that's been built using our own data? And so there were a number of discussions and agreements made, which I'll tell you a bit more about in a second, to make data freely available at the point of almost as soon as it's readily available. It's available to be published. So there's a sort of an assumption now that any large project will make its data available before the publication of any papers in a digestible form, and also that the data, any legacy data that exists in the project will be maintained. And this applies not only to data but also to the things like the ES cells and mice that we've heard about earlier. So there's some web pages here. This is the IKMC website, for example, that you saw earlier. This is an old picture of Europhenome, which was mentioned earlier, and I'll tell you a bit about that. And that's the data from the International Mouse Phenotyping Consortium. And all of these projects basically now have a mechanism whereby the data that they generate goes out public as quickly as it possibly can. So this is a sort of historical list of things that developed out of the genome project originally. So the original agreement was that people like to go to nice warm places to have these discussions. So the original agreement came up. The original discussions took place in Bermuda, and there was an agreement in Fort Lauderdale that the genome sequence should be just put out as soon as it was available, and people could do what they like with it. And there have been follow-ups, so there's the Amsterdam Principles for Proteomics, which is a very similar kind of principle. And the OECD published a report which basically said that scientists have an obligation to make their data freely available. Now, there are limits to this in terms of you have a right to publish your data before you make it available, obviously, and there are certain, you have a right to keep it back for a certain amount of time under certain circumstances. But large projects in particular, which are public projects, have an obligation to make their material and data available as quickly as they can. So this is just to point to some meetings that we organised over the last couple of years. So there are two issues here. One is this pre-publication. So there's pre-publication data sharing and post-publication data sharing. And post-publication data sharing, which is basically once you've published the paper, what do you do with the data, is I think even more of an obligation than the pre-publication sharing. So, okay, you don't want to give your data away before you publish a paper, but once you publish the paper, surely you should make that data available. And the mechanisms for doing that are still not all that clear, because there are all sorts of data types, and where do you put the data is still an open question. So that's the sort of the data freedom thing, and this is really what databases in bioinformatics and in biology are based on. You generate your data, you put it in a database, and it's for the good of the community, and everybody can do what they want with it. And now we'll move on to mouse. So this is we heard, I think Roger was talking about the history of phenotyping, and Paul's been dredging back in the literature trying to find the oldest example of phenotyping. And I think this was the one he found, so he reckoned this was 80 BC as a description of a mouse phenotype. It's the oldest one he could find. So there's a long history, as we know, of looking at mouse phenotypes. And I don't want to go through all of that, but this is the one. So some of these can be actually, attributed to the possible gene mutations. So, for example, the Walser, there are Walser mutants. Some of you may be aware of. And to start this whole discussion off, I've been working for a long time on phenotype, and I want to introduce a few concepts to you, just to clarify the concept of phenotype, because it's not a well-defined concept. Often people use the word phenotype in different ways. So some people talk about the phenotype of a mouse, and they just sort of say, oh, well, it's got this odd, like, for example, it's got the striped face. That's its phenotype. Other people talk about the phenotype as the entire set of everything. And there's a bit of a conflict between the omics concept of everything and the traditional biological concept of everything. So I'm just going to pick up a few terms. I'm hopefully going to use these consistently throughout the talks, although I wouldn't bet on it. So phenotype is an individual observable property of an organism. So it may or may not be abnormal, and that's something that will come up later. But it's something that you can observe and measure, and it's not, well, I'll come to that in a second, but it's basically, for example, the hair is red is a phenotype.
For example?
The hair is red. Oh, no, you won't see it, but yeah. No. The phenome is the set of all genetically determined phenotypes. Now, this is slightly problematic because it's an open definition. The phenome is almost infinitely large because you can always measure more things. But basically what it's intended to do is to express the idea that there's a whole set of things that you can measure about an organism, and some of them are measured as phenotypes. We also came up with this idea of the phenoset, and this was to distinguish the phenome, which is everything, from those phenotypes that you see in a particular individual, because a particular individual has a subset of all the possible phenotypes. And so this is a sort of small concept that maybe, for example, when we measure phenotypes in IMPC, we measure a phenoset for an individual mouse or mouse line. And we were talking last night, and Glauco said, "Oh, there's something else."
Trait, yeah.
Yeah, trait. So what we distinguish is that between the trait, which is the thing you measure, and the phenotype, which is the observation based on that. So the trait is a property of an organism. So, for example, tail length is a trait. Tail length equals 2 centimeters is a phenotype, and that's the distinction. So sometimes that's important, and we'll come back to seeing about that later. We use that.
So let me get this straight.
Yeah.
Okay. So, for example, Brad Pitt is blonde.
Okay.
So that's phenotype.
Yeah.
Color of hairs.
Yeah.
That's trait.
Yeah. So Brad Pitt has hair color. Is
I got this
yeah.
This is exactly where Frege was.
Yeah.
Okay?
Right. Good. So we're philosophically consistent. This is a good thing.
Yeah.
In human, there are other things to worry about. So a lot of what we describe when we talk about phenotypes are physically measurable objects, and there are things that are less well defined. In human disease, we have signs and symptoms, we have syndromes, and these are less well defined, and I'll come back to that. And then we have other obtuse things like gene expression patterns and behavior. Behavior in particular isn't a thing, it's an observation, and it's a little bit more difficult to deal with. So that's some sort of basics on the phenotype side. We're just going to run through a few databases. I'll show you a few databases, and then we'll probably come to the end about that. At the end of that, so I'm just going to name a few databases, and I'm going to show you a few page shots of some of them, just to show you what's in them and what they're like. So Europhenome you've heard about before today. And Europhenome is the database of the International Mass Phenotyping Consortium. It was set up during the Morphia program, the EU program, and it's been developed at Harwell during UMODIC, and it hosts the data from UMODIC and is likely to form the basis for the database for the IMPC as well. A big database in the mouse field, of course, is the Mouse Genome Informatics databases hosted at the Jackson Lab, and I'll show you a little bit about that later. There's an awful lot of information in the MGI database. And there are other useful databases, the Mouse Tumor Biology database, for example, and the Mouse Phenome database, which is also a Jackson Lab database, which has interesting parallels with Europhenome, so similarities and differences, and I'll tell you a bit about that too. So this is the front page of the Mouse Genome Informatics page at the Jackson Lab. And I thought it would be useful to go through this just as an illustration of the kind of data that people have collected over time. So this is their own sort of representation of the data that they cover, and you can see that it goes from the traditional sequences, gene maps, the whole genome sequence of BLAC6, SNPs, gene models, gene products, obviously proteins, transcripts, functional annotation, so that means gene ontology annotations. Orthology relationships with human genes and other genes. And there's phenotype data. And in fact, the Jackson Lab have been very important in developing ontologies for phenotypes, which I'll tell you about after lunch. Gene expression data, although the gene expression data that's held at the Jackson Lab is at the level of tissue expression, it's not at the level of microarray data. And tumor biology. So there's a whole range of data at the Jackson Lab, and you know, this is just to show that there's lots of it. So thousands of data records, more gene records than there are genes, but a lot of information about what's in the mouse. And really, the thing that is most impressive and the thing that makes MGI the gold standard for bioinformatics is the fact that they have a team of curators who go through the literature and extract knowledge. Because extracting knowledge from the literature is one of those things that bioinformaticians can't really do very well. Text mining is still a growing field. There is a field of bioinformatics called text mining, which takes text, tries to extract knowledge from it, and present it back to people. But that's a very difficult problem because it requires computers to understand what's known as natural language, in other words, what people write, and that's actually very hard. You know, brains are pretty sophisticated things, and it's not easy for computers to understand what people write. So the way around that is to get people to do it for you, and this is what the Jackson Lab do. So the knowledge, a lot of the knowledge that's in the Jackson, Mouse Genome Informatics databases is hand-curated knowledge, which you wouldn't, you know, it would actually be very difficult to find even using PubMed, because you know you search in PubMed, you search abstracts and you look for your keywords and you want to find whatever you want to find, and okay, you can use you can work out search strategies, but actually if people read the papers and put the data into a database, that's it's better for you if they do that. And so, yeah, it's not just the same old data. And this is a problem that you do get in bioinformatics. So you can go to 3 different sites, all on related things, and they really have the same data in them. And that's, just frustrating. It's not completely true at the MGI. I mean, clearly they have the genome data, they have mouse gene models, but actually, if you want to look for most of the mouse data that you would want, that's the place to go. We recommend it. So, for example, the papers go into long lists of stuff about genes, and you get descriptions of individual alleles of genes and the phenotypes of those genes, annotated using standard nomenclature, which I'll come back to. And the other thing about this is that you find that you can link together the different kinds of knowledge. So it's important to link together the different kinds of knowledge. So there's no point having a gene in one database, a phenotype in another database, gene expression in another database. You want to have those together, and that's another plus that you will find at MGI. So it's turning into an advertisement, but it's actually a worthwhile advertisement. So the first thing you can do at MGI is just type something into a box and get some result back, and that's obviously what people want. Whenever we ask people what they want, they say, we want a Google type interface. We want to just type something in, get the answer back. There's a long computational gap between typing something simple in and getting an answer back, but everybody wants to do that, and we all understand that that's what people want. And so it does work. I mean, it works reasonably well. You can put in gene names, you can put in phenotypes, you can put in anything that you might expect, and you get something back generally. Or you can click on an area that you want to go into. So these are the main areas that we mentioned before. And you can click on one of these and go into a more sophisticated query. You get pull-down menus, which gives you more detail. So here you've got all search tools, and you've got phenotypes, expression, function, pathways, SNPs, orthology, a list of stuff. So that allows you to be creative with your searches, and you get information about what people at the Jackson Lab are doing. And FAQs, for those of you who are not familiar, are frequently asked questions. So if you don't know how to do something, there's quite a lot of useful information. And so that's mostly data, but we also talked about resources, and you want to find mice. So the traditional way to find a mouse was to go to a page at Jackson Lab called IMSR, or it's not exactly a database, what sort of a database. And basically what they do is they collect together knowledge from across the world about where data are available. So, for example, EMMA, most of the information in the EMMA website also goes to the IMSR. So in principle, you should be able to go to IMSR and find out where a mouse model that you want to find is and where you can get it from. It's not 100% perfect, but it's a very useful resource. Yeah, well, and there's other stuff. And the other stuff consists of, things like, email lists and different kinds of sort of community annotation tools. So one of the things that computational biologists are very keen on, and biologists are not very keen on, is community annotation. So it would be really nice if there was a database out there where you could put your comments in about this gene. So this annotation is rubbish, we have done these experiments and we can show this isn't true. It's actually quite unusual for people to do that, but there are tools on the MGI site that allow you to do that. Various tools. So this is an example of a page that you would get back from one of those searches, a simple search. So this is a gene page from MGI, and just to show you that there's a list of information again. So First of all, the name or symbol of the gene. Now, one of the benefits of the MGI being at the Jackson Lab is that the Jackson Lab is also responsible for standardizing gene names and allele names across the mouse community, which means that the gene names and allele names in MGI are actually correct, which is not always true. So if you go to Ensemble and you start to use it, putting particularly allele names in, you'll have a lot of trouble getting things back. Whereas if you go to MGI, you can get to Ensemble because they can link to the correct gene. You get a genetic map, you get sequences, you can actually download the sequences. So if you want to download the sequence of your favorite gene, you can do that. It'll tell you about homology relationships, so either homologous genes or orthologous genes across species. And it'll tell you about phenotypes associated with that particular gene, polymorphisms known in that gene, function as annotated using the gene ontology, expression patterns, and links to other databases. So really, it's quite a useful hub, protein domains, and other reagents and references. So you really can get a lot of very useful information there. And it's not shown on this page, but the data from the IMPC, International Mouse Phenotyping Consortium, will actually show up in these gene pages eventually in the not too distant future. So the idea is that specialized databases like IMPC, Europhenome, will export a summary of their information to the JAX, so that the JAX will always be probably an integrating point for mouse data. And this is, this is the homology information. So this is just some blow-ups. So this is just to show that this gene has known homologies in certain other species, and that there are also comparative maps you can click on to see how similar they are at the sort of global level, what protein families they belong to, different kinds of protein families, and you can see the gene models, different kinds of gene models. You should be aware that wherever you go to look at gene models, they're almost always automatically generated. So if you go to Ensemble, its genes aren't genes that the people who work on that gene have polished over years. It's what their software gives you back when you put the gene sequence in. And so, again, you're actually getting value by going to this because you can actually see much better curated knowledge. And then there are phenotypic alleles. So there are alleles of the gene which have phenotypes, and there will be different kinds of phenotypes. And here's what you see from the homology. So if you click on the Mendelian orthology tag there, you come up with a list of genes which are homologous to the mouse gene. And this is the tree-fam representation. So tree-fam is a way of classifying genes using phylogenetic trees, which is generated at Ensemble. And it takes a little bit of interpretation, particularly if you're not used to phylogenetic trees. But the idea is that things that belong to a family have a common ancestor. They're all orthologous genes. There are no paralogs in there, in theory. So do people understand the difference between orthologs and paralogs?
Say.
So an ortholog is so we've had this before. So an ortholog is the same gene in a different species, exactly the same gene doing the same thing, to a first approximation, without any gene duplication. Paralogs are the products of gene duplication. So I think we saw a figure, maybe Roger gave it, yeah. So you had the globins. So you had alpha globin, beta globin. They're paralogous genes because they have a gene duplication which relates them. Whereas all the alpha globins are orthologs, all the beta globins are orthologs, alphas and betas are paralogs. It's sometimes useful to know about paralogs. And there are extensions of the concept. So, there are, for example, if you look at the vomeronasal receptor genes, in mouse there are lots of vomeronasal receptors. These are all paralogous, and none of them, strictly speaking, is orthologous to a gene in humans, although they have homologs in humans. So it can get complicated. But the idea of tree-fam is to group together genes which are orthologous, and that's usually what people want to know about because they want to find a gene which has the same function, and generally speaking, orthologous genes have the same function to a first approximation. Yes, sequences. So you can download the sequences. So you can click on that box and say, yes, I want these. I want the genomic sequence or the transcript sequence, or whatever. In different, you can either download it or you can blast it against the genome if you want to do that. And this is the phenotypic description, and this is the polymorphism description. So that's sort of, well, the mouse world to a first approximation. So I'll come back to some of the other mouse databases later. But in terms of where you should go for mouse information, that's the place to go. Humans are more difficult. So obviously we're all interested in how you relate mouse to humans. Where do you go to find human phenotype information? It's a good question. The classic answer is online Mendelian inheritance in man. I'm going to show you a few human databases which cover some of the functionality of the MGI. So OMIM is a collection, it's somewhere between PubMed and a real database. So it's a collection of descriptions of disease states that are associated with single genes. So these are Mendelian disorders only, not QTLs, or not multi-gene disorders. And it's what happens in OMIM is that it's hand compiled again, which is good, but it's basically just a list of stuff that people know about a particular gene or a particular disorder. So it's not organized in, it doesn't use some of the formalisms that databases classically use, but it is really the only source that's available at the moment for human phenotype and disease information that's fairly well organized. So that's the single gene side of things, the Mendelian disorders. This is one of a few a small burgeoning set of gene association databases or data sets. So this is the genetic association database, and this is the other part of that. So if you have a QTL which is associated with a region of a genome, you know, what region of the genome is it associated with? This is the kind of question this database will answer. Gene cards is an integrated data set of descriptions of genes, which allows you to get out from the gene to the information in different species from a central source. So that's actually very useful. But we're now going out from data sets that people have worked on and polished to data sets that people have pulled in automatically. And you should always be aware when you're looking at a database what kind of data is in that database and how carefully people have looked at it. Not because it means it's wrong or bad because it's automatic, but because you need to be more aware of what, you know, the caveats in terms of what's been done. Generally speaking, I think gene cards is fairly good. There are others which are, you know, you can pick up obvious errors in, and that's because people use overly simple ways of taking their data in, which can give rise to errors. But nevertheless, it uses, you know, widely accepted resources as a basis. And I just want to point out another resource which is very important because it's manually curated, and that's the UniProt resource. So UniProt is basically traditionally a database of proteins, although it's expanding now and it's getting more information in it. And again, you know, if you want information about protein function, that's a very good place to go because people look at the data and they decide what to put on to the data rather than stuff being just stuck in. So one way of annotating a protein is to take the protein sequence, search for protein domains, so classic protein domains, and then say, okay, so we know these domains have particular kinds of functions associated with them, so the function of the protein is this, which is, you know, very naive, but there are places that will do this kind of thing. if you can find somewhere where people have actually looked at the protein and know something about the protein, you're in a better world in terms of believing what's going on. And I'll just go through a couple of these things and quickly, and then hopefully we'll get through to the discussion. These are terminologies, so we probably shouldn't spend too much time on them, but just to say that the annotation of human phenotypes and diseases is, is disparate. There are various approaches to naming diseases, and they have different intentions. So mesh terms are basically an indexing formalism, which are quite widely used because they're not unmanageably large. There's quite a good number of them, and they're moderately useful. At the other end of the spectrum, if you like, is UMLS, which is the Unified Medical Language System, which is an NIH development. And NIH is quite well known for having massively complicated projects which nobody has an overview of. And so this is a project which incorporates all kinds of descriptions of diseases, doesn't necessarily link them together very well. And as well as that, if you want to download it, you have to pay. Not good. So there are extremes of naming, and I'm going to come back to naming and diseases in the next lecture, in fact, in more detail. So we have these problems, classic problems. There's the, we like manual annotation, but if we want to produce a manually annotated database, the Jackson Lab employs 60 curators to produce their database. We can't all do that. So many databases have some automatic inference in them. Some have only automatic inference in them, which is not as good as we'd like it to be, but that's a practical solution. There's an issue of resolution. So I mentioned just now that the IMPC database will almost certainly export data to the Jackson Lab, but it won't export all the data because the Jackson Lab doesn't want all the data. All the Jackson Lab wants is a description. And so you lose information. So you have to know where to go. If you want all the information, you go somewhere different to where you go for the summary. Genetic background is not always well described. Again, at the Jackson Lab, it will be generally, but it depends. A lot of literature doesn't describe genetic background well. So if a paper describes an experiment and it doesn't describe what the background was, you're not going to get that information. There's a classic slide which Janan Epic has from the Jackson Lab of the number of ways that black 6 are described in the literature, and I think there are about 100 different ways that black 6 is described in the literature. It doesn't help. And there's a need for standard language in describing things, and this is what I'm going to talk about after lunch. It's very difficult to link together information in different databases if they talk different languages in terms of what they're talking about. If I call, I'm trying to think of a good example. If I call fair hair one thing and somebody else calls it blonde, how does a computer know those are the same thing? It's not easy. So you need to have standardization, and we'll come back to that again. And what we're aiming for, of course, is in the big world, is to take all these resources and join them up so that really you could have a Google. So if you had a single page that you could go to find all the biological information that you wanted, you'd all love that, right? And we all love to do it. But there's an awful lot of work to be done to get that to work. And so these are some of the issues that would need to be resolved, but these are quite technical. Certainly we need to have standards. We need to call the genes the same thing. We need to call phenotypes the same thing. And that's always a hard thing to do. So we actually hold up the knockout mouse data web page is quite a good example of this, because they integrate information from a lot of sites, and they use a standard piece of technology called Biomart, which allows them to link databases together. I'm not going to go into Biomart. I'm not going to go into the details of all this, except to say that they really do actually pull quite a lot of nice stuff together, and it's quite well done. And so, these are some examples of the kind of standards that are emerging or have emerged. Some of them are good for some things, some of them are not necessarily, but things like strain names, ESLIDs, which IKMC has specialized in, genetic nomenclature, phenotype nomenclature, There's a whole list of stuff. I mean, anything you can think of should have standard names, and they don't all exist yet. And even if they existed, they're not used.